let's take a look at the notes for Chapter 13, Section 3, DNA replication. So we have a couple of learning objectives. The idea today is that we can figure out what the two jobs of polymerase is in the role, what role it has in the copying of DNA. So it actually has two jobs. And then also we want to look at the DNA replication for prokaryotic and eukaryotic cells. Please keep in mind, prokaryotic are single cell organisms, usually bacteria, and eukaryotic cells tend to be any cell that has a nucleus, and all multicellular organisms are eukaryotic. But not, we can't do that reversed. All right, so story that we have for DNA so far. So we have Chargaff's rule, right? It gives us this idea of the base pair rules. The idea that the amount of adenine and thymine are always the same. The idea that the amount of cytosine and guanine are always the same. So this gives us the basis for the base pair rules. We then have Franklin's famous number 51, excuse me, and that gives us information about the spiral and symmetrical nature of DNA. Because if you recall in that famous picture, she was able to see that there was a symmetry, that you had equal stackings of those nitrogen bases. And then we know that Watson and Crick come along a little while afterwards, actually around the same time as Rosalind, and using her photograph and using the data from Chargaff's experiment, they put this information together and they come up with this double helix model for DNA. So what is this double helix model? Well, it is a double-stranded anti-parallel model of DNA. Double helix is basically a fancy way of saying like a twisted up ladder. So I have two molecules of DNA, one on the left, one on the right. They hook up at the middle and then they get spun around. And an anti-parallel means they run in opposite directions. We know that the backbone of DNA is held together by covalent bonds. And it is the sugar, the 5-carbon sugar, and the phosphate group of the nucleotide. We know that the DNA base pairs, AT or CG, are the rungs of this twisted up ladder, and these are held together with hydrogen bonds. And we know that DNA has 3 jobs. The DNA jobs are, number 1, it is meant to store. 2, it has to copy, and 3, it must transmit genetic information. So DNA will store genetic information, DNA can copy genetic information, and DNA will transmit its genetic information to the next generation. So this is everything that we know so far. So let's talk DNA replication, right? We've been working on this all week, so let's talk about it finally in a formal manner. I do want to remind you that DNA replication is going to take place during the S phase of the cell cycle. So we have mitosis and meiosis. These are the 2 different ways in which we can replicate a cell. Mitosis, again, is when we make body cells, we make replication cells, or repair cells, excuse me. This is also the time that we are making asexual reproduction cells. Meiosis, on the other hand, is for purposes of making sex cells or gametes, but either way, you are having cell division. And before you have any kind of cell division, you must have DNA replication. DNA replication is just a fancy way of saying it is the process by which DNA copies itself. Remember, if you are eukaryotic, this is happening in the nucleus before the cell can even think about dividing. Before mitosis, before meiosis, during that interphase stage of growth, during the S phase, which is the synthesis phase, we are going to be creating a brand new, complete set of chromosomes so that the new cell being created gets a full set of chromosomes. So we are going to be able to transmit all of our DNA, all of that code to the next set of cells or the next generation, depending on if we're just doing repair or if we are doing reproduction. All right, so there are some enzymes that we have been talking about that are prominent in DNA replication. In fact, these guys are kind of like the stars of the show. We have helicase. Helicase is the breaker. Helicase unzips the DNA strand, and it does this by breaking the hydrogen bonds down the center of the double helix. We have polymerase. Polymerase is our builder. Polymerase is responsible for building up the brand new DNA strands. RNA primase is like the boss. It tells the builder, the polymerase, where to start and what to do. So kind of like your mom is the boss of you and she tells you what to do and how to start up your projects, your jobs. RNA primase is kind of like the boss of polymerase.
Here.
We then have ligase, which is a binder enzyme, and this binder enzyme is going to be gluing new DNA fragments together. So we'll talk about what that means in a moment. All right, so step one, DNA replication. So step one is going to involve helicase. Helicase is going to unzip DNA down the middle by breaking the hydrogen bonds and creating a replication fork. In the picture underneath, you see that sort of like whitish pink blob right there, kind of where the Y is made. That is supposed to represent helicase. So helicase literally goes down the molecule and breaks apart the the two sides of the DNA. All right, so in a prokaryotic cell, so again, these are bacteria. Bacteria have fairly simple DNA, and they tend to have only like one strand of DNA inside them at any one given time. They have circular DNA, and this means that you have a single replication bubble. So again, that means that you would have two replication forks. So you'd have polymerase pop on, and it sort of starts unzipping in two directions, okay? So that is one way that prokaryotes do their DNA replication. In eukaryotic cells, however, like the ones that we have 46 chromosomes, which means that we have like 46 strands of DNA in our body, in every cell of our body. Our DNA is linear. Eukaryotic DNA is linear, and it's very, very, very long. So what happens is lots of different polymerase jumps on, and it's kind of like having a broken zipper, if you want to think of it that way. It just starts unzipping itself in multiple replication forks in multiple parts on any strand of DNA at any time. So you're going to have many replication bubbles because, again, if they start on one side and went all the way to the other, it would just take entirely too long. So we just kind of do like multiple attack vectors. All right, DNA replication step 2. So now we have RNA primase. So the primer, the boss, before the strands can form, the boss, the RNA primase, comes along and it sort of starts to give instructions. It says, okay, this right here is where we're going to start. So you can kind of see it right there in purple. It says RNA primer. That right there is going to tell, again, the polymerase, which is coming next, okay, this is where we want to start replicating. And then you can see these new nucleotides being added on. How are those new nucleotides being added on? Well, that would be step 3. Now in step 3, we have this DNA polymerase. And again, that are these like, sort of like purpley bean-looking things. They find the RNA ligase, the RNA primase, which is where you kind of see that gap right there. And then they start adding on those new DNA nucleotides, okay? Brings us to step 4. Now, step 4 is all about replication. Replication can only move in one direction. Remember, DNA has a direction from 3 to 5 prime or 5 to 3 prime. That is the direction of DNA. Replication only goes from 5 prime to 3 prime. So you can see right here, the replication is going this way from 5 to 3. Down here, I am going from 5 to 3. 5 to 3. The problem is that I have a leading strand on the top. So the 5's right there and it's moving in the 3 direction, no problems whatsoever. However, I do have a problem here on this bottom strand. This bottom strand is known as the lagging strand because, again, if you're going from 5 to 3, you end up having these little tiny fragments of DNA because the primer doesn't really know where to go. Not that the primer doesn't know where to go, it's just that it keeps attaching itself. So you end up with these little tiny fragments called Okazaki fragments. And that is why it's called the lagging strand, because it takes a little bit longer to make that particular strand. Step 5. DNA ligase comes up and seals up all of the gaps in the DNA molecule, like the Okazaki fragments. So all those little openings that were there, where the primase used to be, the ligase jumps in and then glues the DNA together. All right, and then Shazamo, DNA replication is complete. What I want to point out right here is that now I have my original, which was 2 strands, both in red, and now I have my copies. But notice the picture of the copies. One strand in the copy is red, exactly like the original, and then I have my new strand, which I built, which is orange. So I end up with 2 identical molecules which are formed from the original. But again, these look slightly different. So are they really identical? Genetically, yes. But we do have something called semi-conservative model of replication. What that means is, and by the way, this was an idea presented by Watson and Crick. What this means is that 2 strands of the parent molecule, as they separate, each acts as a template. So as they separate, you get new nucleotides built on to each one of those. And we get a brand new complementary strand from the replication process. So what this means is that my new DNA is made up of one original copy of the DNA from the parental DNA and one brand new strand that the enzymes just put together. Okay, so because we have one from before and one that's brand new, we say it is semi-conservative. The semi-conservative model means that every time I replicate DNA, the new DNA is only half new because it's one old strand and then we built a brand new strand on top of it. All right, so we're doing all this building, and as you guys know, if you've ever built anything, sometimes mistakes happen. DNA polymerase is our builder, and when it's going through and it's building, it makes some mistakes. So for every 10,000 base pairs, it makes a mistake, approximately, okay? And that doesn't sound like it's that bad, but remember, each strand of your DNA is millions and millions and millions of nucleotide or base pairs long. So 1 in 10,000 might not sound like that much, but when you're talking about millions and millions and millions, it is quite a few mistakes. Here is the second job that DNA polymerase has. This enzyme can proofread. So it will go through and it will proofread its work, and it corrects those mistakes. So that is a very, very important second job that DNA polymerase has. So first job, of course, is to build the new DNA, but the second job, and maybe more important, is to proofread the DNA. Once we go through the proofreading process, we go from 1 in 10,000 base pairing errors to 1 in a billion base pairing errors. Now, there's still some errors, but instead of 1 in 10,000, it's now up to 1 in a billion. So that's a lot fewer errors because of this awesome proofreading job that the DNA polymerase does. So, but there's still mistakes. So that leads us to, what are these mutations then? A DNA mutation happens whenever there is an accidental change of a base. So maybe, you know how we said, well, A and T always go together, C and G always go together. Sometimes there's a mistake, like this 1 in a billion mistake. And instead of pairing an A with a T, maybe instead it's going to pair an A with a C. I'm making this up. Wow, okay, that's cool, but it's a mistake. That DNA mistake, that DNA miscode, is what we call a mutation. Now, people always think that mutations are bad. Sometimes they are, but sometimes DNA mutations can be a good thing. And the reason why is because it is a source of genetic variation. Remember, genetic variation is what drives evolution. Genetic variation helps with the survival of species when there is a change to the environment. So some genetic variations aren't great, and maybe they're not wonderful for the species, but some genetic variation is amazing and it is great for the species. And again, that is the driver of evolution, is genetic variation. So DNA goes in, proofreads the new DNA polymerase goes in. It will proofread our new DNA and catch most of these mutations and fix them. But it's not going to catch them all. So there are going to be slight miscodes in everybody's DNA. By the way, this is why identical twins, the older they get, the more different they become, because each individual then has slightly different mutations in their DNA. So, we've all seen superhero movies. We've all seen sci-fi shows. What are some other causes of mutation? We know this. We've seen movies. We've seen superheroes. There can be environmental agents that cause genetic mutations as well. Again, I'm a huge nerd when it comes to like superhero type movies, and I kind of love mutations. Now, let me just say this. Most of the mutations they have in superhero movies wouldn't actually happen. Most of the time, that kind of stuff would actually lead to, like being dead, kind of like when Hulk got gamma radiated. He would die, but not turn into a green monster. But hey, whatever. Science fiction being the operative word. So, UV rays can cause some mutations. We see that a lot in plants. Chemicals can cause mutations. We see this a lot in frogs. And X-rays can cause mutations as well. We see this. We actually used to see this a lot in X-ray technicians, in the sense that they would get cancer and things like that. Until we figured out that you wear that big lead sheet when you get your X-rays, and then it can't go through those. Okay, so that leads us to the end of our 13.3 DNA replication notes. As always, if you have issues or questions, please feel free to ask.